All right, welcome everyone. My name is Tom Lowry. Thank you everyone for being prompt. We're right at 12 o'clock, so we're going to go ahead and start. This is a 45-minute session, and listening to me for 45 minutes is a risky proposition, so if you start to nod off, don't worry, I won't call you out. So I'm Tom Lowry. I'm the Chief Scientific Officer from T2 Biosystems. I also happen to be the first employee that was hired there about over 12 and a half years ago. And have been involved in developing the technology and pushing forward the clinical work and work with the FDA and so forth. So I'm excited to talk to you today. I'm going to talk about quite a lot of different topics here. I want to share some perspective on direct from blood diagnostics based on my experience in this field. I want to share some new clinical utility data from T2 bacteria that I think you're going to find very interesting. Also share some new clinical early feasibility data demonstrating some of our new panels and talk about our other panels. But before I dive into the details, I want to share some motivation that we have for us here at T2 Biosystems. I think a lot of folks, you do as well, for the need for direct from blood diagnostics. So there's a lot of patients that pass away without knowing what pathogen is infecting them, and therefore not knowing if they have appropriate antimicrobial therapy. The person on the left happens to be my brother, who passed away in the mid-70s, he had an abdominal sepsis infection. They never figured out what pathogen was infecting him, therefore didn't know if he was on appropriate therapy. The person on the right is a good friend of mine and a father of two employees at T2, Mark Triscutt. He was also the VP of R&D at Instrumentation Labs. He had defeated cancer, and then just last year he had a pneumonia infection that progressed and disseminated throughout him, and they never found out the pathogen before he passed away. And so they didn't know if they had him on effective therapy. So it's a really interesting study that came out in JAMA that actually highlighted this. They went in and they looked and asked the question, are there avoidable deaths in sepsis? And it turns out there are. And in fact, there's deaths that are people who are dying from sepsis and they have suboptimal care. I think all of us as caregivers and folks in the industry are trying to help minimize the instances of these. So one of the conclusions of the study was, of those patients with suboptimal care that were deaths in sepsis, about half of them could have been prevented. And we broke them down into these different categories. So delay in antibiotics, we all know about the urgency of getting IV antibiotics on quickly, delay in source control, and then even almost 25% of them actually had inappropriate empiric antibiotic therapy. And so part of our mission at T2 and any direct from blood diagnostic is to help minimize these cases of suboptimal care. So to talk a little bit about why that's the case and why there's this important interplay between diagnostics and therapeutics for patients who are suffering from bloodstream infections, I wanted to show this chart where the top row is the antibiotic therapy regimen and the bottom row is the blood culture and diagnostic regimen that we use. So for decades, we've been relying on getting a positive blood culture from a patient to help identify the pathogen that's present and then identify the resistance markers and or the susceptibility. Now that information, as it flows back to the clinicians, it influences the care. Because there's this significant initial delay of information, patients that are suspected of bloodstream infections are put on empiric IV antibiotic therapy. And then once you have a gland stain, you make a change. Once you have species-directed therapy, you make a change. So what's staggering about this is that patients who are at risk and suspected of sepsis, who have a blood culture drawn, get about a positivity rate of about 10%. But we're treating about 60 to 70% of those patients with IV antibiotics, so a very high treatment. However, and the data is very clear, there's a great meta-analysis that looked across 70 studies. Only about half the patients that are on empiric IV antibiotics are not on effective therapy. So even though we're using so many IV antibiotics, trying to hit so many patients, we're not able to get the patients on the right therapy for the organism that they're infected with. Once the species ID information comes back from the lab, the data are very clear that you get above 90% of the patients on effective therapy. And what's really fascinating and tragic is if you have some resistant markers in your infection, like carbapenemase resistance genes, close to 10% of those patients actually pass away before the AST result even comes back. So we have a real problem with the speed to diagnosing these patients and the dependency of knowing if your patient's infected and knowing what they're infected with in order to help inform appropriate therapy. We're all familiar probably with these studies that have demonstrated, there's been a few, a recent one that came out as well, every hour without effective therapy, if you have a bloodstream infection, your odds of death go up by 4%. Every hour without effective therapy, if you're in septic shock, your odds of death are going up between 7 and 8%. So that time requirement is really important. Blood culture is helping you in that 12-hour range. So a great study just came out that looked at blood culture use and the time to results. Median time to result is about 24 hours. Mean time to result is closer to 2 days. And then species ID center, median time to result is about 2 days, and then mean time to result is 3 days. So our mission at T2 is to help provide the information earlier than is possible today with blood culture to help inform effective therapy and other treatment decisions when basically they're relying on empiric therapy. So that's the whole premise of culture-dependent, independent molecular diagnostics. And for over a decade, companies and technologies have been trying to provide these kinds of tests to folks like you in the laboratory and clinicians. So culture-dependent molecular diagnostics require a positive culture. It's very well documented literature that a single set of cultures has a sensitivity of about 50 to 73%. And that's why the guidelines recommend that you need to do 2 to 3 sets of blood cultures to get your clinical sensitivity up above that 95% range. Now, any post-culture diagnostic test is going to be limited by the actual sensitivity of the culture and the time to result. Once you can get that result, that positive culture, then you can go and do your post-culture workup. So there's a lot of effort to realize that. The history here, there's been a lot of efforts trying to develop these types of direct from blood tests. The first company in the area was Roche. They developed the Septifast test, a manual test. It's CE marked. It never was available clinically here in the United States. There's 2 other CE marked tests out there that have some pathogens for direct from blood. There was a really great panel from Abbott Aritica that was available, extremely comprehensive. That was pulled from the market, unfortunately. They never were able to get FDA clearance. And then there's a few companies that actually announced that they were going to go direct from blood and then went post-culture, or they took a post-culture system, explored if they could go direct from blood, and haven't demonstrated that yet. And there's a few companies, I haven't got all of them up here, that are in the development phase trying to get direct from blood. So there's a lot of efforts to get there. There's only been one company that's got an FDA clearance, and that's T2 Biosystems, and now is in clinical use in the US. And you may ask why. Why is it so hard? Why have so many people tried and not actually achieved that? Well, I've lived it for 12 years, and so I can tell you why. The first one is related to you've got to have exceptional clinical or analytical sensitivity. You have to get down to that sensitivity that's equal to or better than blood culture for intact pathogen cells, and that's important. You need to be able to detect those cells down near 1 CFU per ml to have robust performance. Now, this is really important. In order to do that, you've got to make sure you quantify your cells in an accurate way without bias. If you measure your cell count in blood after you put it in blood, you're going to get an extraordinary bias and underestimate how many cells are there. So there's very specific, rigorous methods for measuring your CFU count based on the solution that you spike into the blood. The other areas that are really important is you got to have sample stability. If you don't have sample stability, that introduces a lot of bias into your studies. So you got to have this extraordinary sensitivity. But not only do you have to be extraordinarily sensitive, you have to have a very high specificity. I'm going to take you through a few platforms where that actually was the reason why they couldn't be deemed safe and effective for use here in the US. So very high specificity as well. So let's go ahead and compare some performance data for platforms that have either announced that they're going to try to go direct from blood or have been direct from blood. So you can see that analytical sensitivity needs to be in between 1 to 15 CFU per ml is our general rule of thumb for a direct from blood test. Your specificities need to be, or sensitivities need to be north of 90%. And your specificities on a per result basis really need to be close to that 98% level. The Roche Septifast platform and the other CE mark platforms they're a little bit higher on your limits of detection, and then their sensitivity and specificities are also a bit lower. The Aritica platform, which had really impressive analytical sensitivity and impressive clinical sensitivity and specificity, that system, though, the challenge there was even at 85% specificity, it generated a lot of positives that didn't correspond to blood culture, and so didn't know what to do with those results, even though a lot of them were proven to be clinically useful. Accelerate had announced years ago they were going to go direct from blood. If you look at the 510K summary, their limit of detection is actually 1 million CFU per ml. So that's a significant gap there in terms of direct sensitivity. BioFire platform, they have had some grant funding to go direct from blood. There's still a gap there that needs to be bridge in sensitivity. And I think the Carius platform is a great platform, and it's in the space of direct from blood. They're detecting cell-free DNA, have extraordinary sensitivity. They identify a lot of things that blood culture doesn't identify. If you compare that to blood culture, though, you get a really low specificity. And so that's one of the challenges here, is you've got to thread this needle of agreeing with blood culture while yet most likely being more sensitive to blood culture. So a lot of people, when they think about our panels, they say, your panels just aren't comprehensive. So we've taken a fairly steady approach. Our first panel was a 5-member panel for T2 Canada. The second panel was T2 bacteria panel. And we've done that for a couple different reasons. And one of the benefits is we've been able to manage the performance of the panel and understand what it takes to get FDA clearance, what it takes to get robust clinical performance. We have a T2 resistance panel I'll tell you more about, but now we have enough data in-house that we can deliver a panel that's comprehensive of 99% of all pathogens in bloodstream. And this is the target panel here. And a key part of this panel is what we call pan-gram positive, pan-gram negative channels. So those are available on post-cultures. When you take that pre-culture, it's really tough to get the requisite sensitivity and specificity, so you're only detecting intact pathogen cells at a high degree. And so we're excited we have the detection data now that supports the ability to do that. You'll see there's some genus level channels, species level channels, as well as resistance gene channels. So some of these formulations we already have on the T2Dx, and now we have a master formulation that we have demonstrated feasibility around for a future panel that will be 98% coverage. A fascinating dimension on this, and we'll see over the next year, you'll see research data coming out from customers of ours who are actually looking at using our panels, not only direct from blood, but also post-culture. So you can envision a molecular platform in the future that provides both types of results. But what do we have to work with today, particularly clinically, are these panels here. They've been deemed safe and effective, the two on the left, so T2 Canada and T2 bacteria, for use in patients today. And I think that's a really important thing, because the question then becomes, can we see clinical utility in these types of panels, even though they may not be comprehensive? So I'm going to take you through a lot of clinical utility on those panels. Then you have the T2 resistance panel. I'll take you through that. That'll be available in Europe by the end of the year. And it's received breakthrough designation from the agency, so we're working on a timeline for FDA clearance. And I'll take you through the other two panels as well. Just to orient you, if you haven't seen our instrument, you can come over to our booth and check it out. You can load up to 7 cartridges at a time in this instrument. It's a random access fashion, fully automated, sample to answer. And so fairly easy to use. Use a 4 ml EDTA vacutainer. Minimum blood volume is 3 mLs to attach to the cartridge and load on the instrument. The instrument's got a touchscreen user interface. Everything that's used for a sample is loaded on with that cartridge, and any waste comes off with the cartridge. So there's no other types of shared disposables or reagents. How does it work? So what the instrument does is it takes a 2 ml blood volume, and it actually selects for intact pathogen cells. It does a simple centrifugation step to take all the debris. And then what we do that's a bit opposite from a lot of molecular tests is we discard the supernatant, and we want to actually just look at the intact pathogen cells, so we keep the pellet. And we work in that complex matrix for the remainder of the processing. So we do an amplification step in this semi-solid material, and then we take the amplicon from that, and we detect it with our T2 magnetic resonance detector. So the benefit here is, because we're using a detector that's non-optical, it doesn't matter what kind of interfering substances or debris, other things that are present in the sample. The amplification detection is very robust against that. So for example, the T2 bacteria panel, there's one interfering substance, and that's an MRI contrast agent, because we measure an MRI signal. But whether it's antibiotics, lipids, or whatever else it might be that's in the sample, it doesn't interfere with the test. So let's just jump into, to start off, T2 Canada. This has been out on the market for about 4 years. 5 species of Canada, sensitivity and specificities are listed there. Just to underscore, when this panel was cleared, it created a new product code at the agency. So it's a brand new type of test for direct from blood detection of fungal pathogens. So when we had our first accuracy study come out, there was a lot of discussion about that. It demonstrated great accuracy in terms of sensitivity and specificity. The thing that people really didn't like about it was there weren't that many blood culture positives. So there were 1,500 patients in the study. I think we had a total of 7 blood culture positives. And that's a challenge with these types of studies. The agency set these studies up so they can look at the accuracy of the test, and then it's up to us afterwards to demonstrate clinical utility and more data around prospective patients. So this study shown here was one that Neil Clancy authored. 152 candidemic patients. And he actually demonstrated the clinical sensitivity in the larger patient cohort of that size of 89%, which is a very nice validation of the trial design, where we had originally seen a 91% sensitivity. The other interesting thing that came out of this study was actually looking at how the test performs in patients who are already on an antifungal. And that's where you'll get often a culture negative T2 positive. And so the utility of tests for those kinds of patients is fairly interesting. So I won't spend a lot of time going into this dimension, but if you're using these types of tests, I think it'll hold true for other types of culture-independent tests, is that they can come up positive for patients that have deep-seated infections. And so there's a great study, the first one that I list there from Dr. Maiken Arendrup in Denmark, where she showed that the best diagnostic tool set for invasive infections for Canada is a combination of culture and T2 Canada. And I'll refer you to that to read more about that. There's been a variety of other cases. This kind of attribute of these tests can present challenges to a laboratorium because you have a culture negative sepsis case and you have a positive. In fact, in our first accuracy study, that's the patient on the bottom here, we had a patient that was negative across 12 blood cultures, positive by T2, so it was counted as a false positive. However, they actually had a biopsy from a sterile site that was cultured positive. And that was the first case of candidiasis that the test detected. And that was the beginning of building of this data, that this attribute of these types of tests. So it's something to be aware of. A lot of times customers use that T2 positive, culture negative, to go and look at other potential sources of culture samples and evaluate the patient with imaging, and then they'll find a deep-seated infection. We don't have time to go through all the other studies on Canada, but just to wrap up on the Canada side before we go to bacteria, so there's been a variety of studies verifying that yes, using a test like this can get your patients on effective therapy far faster than the standard of care. And also, prior to implementation, the second bullet here, they were detecting about 57% of their cases, and after they implemented them, they were detecting 93% of their cases. So being able to catch these infected patients with a test like this is a significant attribute that attracts folks to it. A fascinating side observation has been in these patients, ocular involvement of Candida decreases. It may be related to catching it earlier and you don't get as much dissemination of infection. There's been a variety of financial studies that have been reported as well. We won't go into all the details today due to time, but there's two different models that people are using this type of test for. One is to withhold antifungal therapy in the right patient population for that, those types of patients. They're seeing significant cost savings. The other model is to actually find patients who are really, really sick and get them on appropriate therapy early. And we've had a customer who's received a quality award from their hospital. There's a poster in 2017 showing that they got a significant length of stay and annualized savings. So now we'll shift gears to T2 bacteria. We got that cleared about a year ago, and then we started our sales effort and moving into installing and implementing it. It was a big accuracy study, just got published in the Annals of Internal Medicine. It was just that, an accuracy study. We were not looking at clinical utility in that study. We're really excited that we got very similar performance for the bacteria pathogens as we did for Candida. After we got the Candida clearance, a lot of folks came to us and said, that's really fine and good, but I don't think you can do that for bacterial pathogens. And so that was a big focus for us, is being able to develop a panel that had the same kind of characteristics for these pathogens. Now, for you microbiologists, I don't think we should call it a bacteria panel. We should probably call it an escape pathogen panel. These are the pathogens that covers 90% of infections caused by escape pathogens. Those are known to actually escape, are most likely to escape IV empiric antibiotic therapy. And so we felt like those pathogens would be the most valuable initial set of pathogens to deliver in a test like this. Another sort of discussion point we've had a lot with folks before the data came out was, well, you aren't going to see the same time savings because E. coli cultures really fast. So you can see there on the bottom right, there's some time charts. So the 5-hour turnaround time for bacteria. In our study, the turnaround time on average was about 70 hours for species ID, and that's based on a MALDI result that's coming post-culture. And so you can go to a variety of other studies. If you look at the median, that shifts up about a day. So you're in the 48-hour time if you're looking at the median and not the mean. And so there is a significant opportunity to get these results a lot faster to patients. And the other attribute that was really interesting from the accuracy study was the test is detecting over 3 times more patients infected than the paired blood culture. So in other words, we got a T2 positive. How many blood cultures were drawn from that patient to get a positive blood culture? And you get about a factor of 3 there in terms of enrichment. So that provides a value to be able to catch these infected patients a lot sooner because you're not having to go back and do multiple draws. So in this study, we did have access to antibiotic information. So there's some papers in peer review right now that go into this analysis. There's been a few presentations on this. The question was asked, how many patients at that time 0 had effective antimicrobial therapy? And so the answer is about 31% of them had effective antimicrobial therapy. Now in this study, the results were not shared with the clinicians. And so we're looking at just the standard of care here. At time 24, we have about 80% on effective antimicrobial therapy for these patients. Now this is a mix of a bunch of different sepsis patients. Some had had cultures drawn 3 days before. Some in the study, this was their first culture. We can see there's a significant opportunity to get these patients on effective therapy faster. And I'm going to jump into a lot more data here, because the goal here is to get this to 100% within 6 to 7 hours rather than waiting a day or multiple days. The biggest area of interest for use of the T2 bacteria panel is in the emergency department. This is a study that's been presented at a few conferences and also is now in peer review. This is performance of the test just in 2 different emergency departments. You can see 100% sensitivity and 98% specificity. Emergency department actually is a very convenient place to do these types of studies because you don't have patients on a lot of prior therapy, and so you don't get as many discordant results with culture. One of the fascinating things that we observed in this study is of the patients who were T2 positive, they did an interesting analysis, and the analysis in this was to say what pathogens identified and were they on effective therapy and how long did it take for them to get on effective therapy with blood culture. We saw that for 26% of the T2 positives, they could have been on effective therapy 16 to 67 hours faster if the T2 result was available. And then 35% of the positive patients would have been on actually species-directed therapy. And so these kind of analyses, particularly for folks in antimicrobial stewardship programs, are pretty easy to do because they're already making these therapy adjustments based on the species ID from blood culture. They have the T2 result from the same patient that was used observation, and they say, oh, wow, we could have actually, if we knew it was Staph aureus, could have made it early. And I'm going to take you through a variety of case studies to clarify that. So a big effort of ours is with each one of our customers as we partner with them in helping understand how to use the test and the patient selection criteria. And that's an important question because today, patient selection criteria for molecular tests is I have a positive blood culture and I know to run it. But when I have all patients being run with blood culture, you certainly want to go and use a test like this on all those patients. There's a lot of indiscriminate use of blood culture today. And so we have a team of clinicians led by Dr. Sandy Estrada that partner with the customers to go in and say, all right, what patients are at risk? And we look at the epidemiology of the patients in the hospital, look at different criteria that can be used to control the order set. So on the left, you can see different things like QSOFA scores, lactate values, PCT values, and then also looking at different patient populations based on their underlying physical condition. And this allows the hospital then begin to roll out their test and interventional use in a way they can understand who's going to benefit and to analyze it and then work on evaluating other patient populations as time progresses. The other dimension is what do I do with a result? And so we, based on the early adopters of T2 bacteria and our own clinical team, we've developed some algorithms for what one should consider in terms of clinical treatment and what kinds of decisions they could make early. And I'm not going to go through all the details, and this is not on the final exam, don't worry about that. But it's something that we've been really excited about as people begin to see the value of these direct from blood tests. And one of the key things is when you get a species ID result, you go and do what you do already today, and that's you go to your institution's antibiogram. So when you go to your antibiogram, that tells you which, in your institutions, which therapies work for which organisms, and based on the risk profile of your patient, you then can go ahead and initiate antibiotic therapy. So that's a key step in using the T2 bacteria panel. So you may say, okay, what kind of studies are going on? When are we going to see some results? So there's over 12 studies right now that are investigator-initiated studies that are going on looking at the clinical utility of the test. We have 5 investigator-initiated interventional studies going on. Within a couple months, we'll have another 4, so it'll be a total of 9. And we're doing everything we can as a company to partner with folks to help explore the test's use in different areas and to demonstrate value. And as these studies have been progressing, we've been able to look at the clinical cases and start getting a good feel for the data, and I'm going to take you through some of that data today. So I got data from 7 interventional studies. These are mostly in the ED, but there are also some studies that are in the ICU. And of the cases that have been evaluated for clinical benefit, right now we're seeing about 42% of the patients have demonstrated clinical benefit for the use of the T2 bacteria panel. And they fall into three categories. One category is T2 positives that can actually get your patient on effective therapy faster. The other category is T2 negative that can be used for de-escalation. And the third is avoiding premature discharge and readmission to the emergency department. So the bar chart on the right, or actually before I go to that, so you may ask, well, what about the positivity rate in these studies? So the average positivity rate we're seeing is 16% for T2 bacteria, and that ranges from 13% to 35%. That depends on your patient population, depends on how you're using the test. For the studies that are using the T2 positives, and they're actually saying, I want to use the T2 positive to get patients on effective therapy faster, they're seeing that 28% of those patients could benefit or are benefiting from faster effective therapy. And that's lined up pretty nicely across the studies. Now there's 3 of these 7 studies that are focused on actually using the test for de-escalation, very specific patient populations, particularly for Pseudomonas and Staph aureus. They're seeing about half their patients are benefiting or could benefit from the use of T2 bacteria for that de-escalation. So what I thought I'd do today is I'm going to take you through 5 example case studies to get you a flavor for what this kind of information is doing. And I think this is going to hold true for direct from blood diagnostics can deliver results at this sensitivity and specificity. So the first case study is an anti-pseudomonal de-escalation. So in this case, the site here, they're using the T2 bacteria panel interventionally, so they're using the results to treat the patients. This patient was receiving dialysis, they were unresponsive and developed sepsis, and they were put on a very common empiric IV antibiotic therapy of vancomycin and piptazo. T2 bacteria came back positive for Staph aureus, negative for the other pathogens. Based on their epidemiology in their hospital, they decided that it was appropriate to de-escalate the piptazo. Blood culture came back positive for Staph aureus at a later time. And so the case analysis here is that because the patient was at risk for nephrotoxic therapies, it was valuable to de-escalate the piptazo, and the patient was sent home after a total duration of 6 weeks of therapy. Let's go to another case. This is where T2 bacteria also was being used interventionally. This is a patient, nursing home patient, the infection started as pneumonia, and they were put on vancomycin, ceftazidime, and clindamycin. Okay? T2 was positive for Klebsiella pneumonia, negative for the other pathogens on the T2 panel. And the blood culture results, this was a negative. This is a case of blood culture negative sepsis. So what they did with that T2 result is they actually initiated meropenem on the patient, and that they were very encouraged by is within 1 day they had white blood cell count that dropped significantly. And they felt like that was a good sign that they had the patient on the right therapy. After 7 days of meropenem therapy, they were able to discharge the patient back to the nursing home. So this was one that the site got very excited about because they were able to catch an infection that they would have otherwise missed. In this study, the T2 bacteria panel was getting used observationally, and they were doing analyses with that data after the fact to see what kind of impact they could have observed. This is an emergency department. Patient had signs and symptoms of a UTI, and the T2 and the blood culture draw were taking. In this case, because they weren't using the T2 draw, they were batching at the end of the day. That's why you see an 11-hour turnaround time for T2. It was positive for E. coli. Now, they actually drew a blood culture draw. They also did a urine analysis, and they put the patient on IV dose of ceftriaxone. In this case, because the urine analysis came back negative, they discharged the patient. The patient left the hospital. The next day, the patient actually came back to the hospital with fevers and chills. Symptoms were worsening. They readmitted the patient. So after they did the analysis, they felt like they could have used the T2 bacteria test to help avoid an unnecessary discharge of the patient because the blood culture result just wasn't coming back fast enough to avoid that. This is case study number 4. This is a case of ephesium, a tragic case here. So this patient, ICU patient with ARDS, had signs and symptoms of infection on day 0 of this case. They took the T2 draw for T2 bacteria. This is an observational only case. It was positive for ephesium. The patient actually started to rebound back at the end of day 0, and so they didn't have access to the T2 result based on the rules of the study. On day 4, the patient crashed. Another blood culture was drawn, and that blood culture actually came up positive on day 5. The patient had passed away, and they actually never were able to get the patient on therapy that would have addressed ephesium. And so I think the case analysis here is pretty obvious. We don't know if it would have avoided the death, but we do know that it could have been better clinical care to have the patient on effective therapy for ephesium. And the last one I'll show is a Staph aureus case. We've seen several like this just because of the prevalence of Staph aureus. In this situation, the patient had a history of cancer and COPD, came to the emergency department with fever and chills, sepsis was suspected. They ordered blood culture, they put the patient on IV antibiotics. The T2 bacteria, once again, observational only. And actually something interesting with this site, the way they're using the T2 bacteria panel is they're using their lactate and their PCT, and they have thresholds defined for those, and if they're above a certain threshold, then they run the T2 bacteria panel. So in this case, they ran T2 bacteria. It was positive for Staph aureus, negative for the other panel members. Blood culture came back. They got the species ID 36 hours later based on their antibiogram. They actually moved to a therapy that the patient was not on. And so their analysis was that they could have had the patient on that therapy 30 hours sooner if they were using T2 bacteria clinically. So hopefully that gives you a flavor for the types of clinical use cases that this type of panel is having, what we're observing. There's about 24 peer-reviewed studies on the clinical uses of our tests. Just to highlight some takeaways about direct from blood culture, or direct from blood and culture-independent tests that we're seeing with our technology, is that, yes, it's faster than blood culture-dependent methods. It does enable a dramatic reduction in time to effective therapy. Sensitivities come into that 90% range. Specificities come into that 95 to 98% range. You get very nice NPVs for the panel members. And then PPVs depend on where you're using the test. That's something you need to think about. And then there's a lot of papers that are showing T2 Canada can really reduce costs for reduction in antifungals and faster time to therapy. And then today we're at the beginning of the T2 bacteria data. At least we're seeing now that it's useful for these three different classes. You can get patients on effective therapy faster. You can get patients avoid unnecessary discharge and de-escalate therapy. You may wonder, okay, where are the T2 systems? This is our current placements. We have 97 placements, 170 hospitals are implementing or have implemented our tests into their workflows. You may say, well, what about your customers? Here's some of the customers that have been publishing. Some great leaders in the field have been publishing with T2, and we're really pleased to be partnering with them in helping improve their patient care. So our last little bit, I'm going to actually just switch gears and talk a little bit about T2 resistance and then the other two panels. So the T2 resistance panel, this is a comprehensive set of resistance markers that are commonly present in bloodstream infections. You have your gram-positive resistance markers, your CTXM to cover ESBLs, and then you have a lot of carbapenemase resistance genes. So this panel here, we announced it earlier this year. We're excited that the FDA gave us breakthrough device designation. We're working with them on the details of the clinical trial. So we haven't announced timing for the FDA cleared product. We will be in the market by the end of this year. We've already got initial preclinical data from 2 sites in Italy that was presented at ECMID. Same kind of analytical sensitivity as our other tests, same workflow. There was a question after T2 bacteria, can you do this for resistance genes? The answer is yes, you can. You can get the same kind of performance. And so we're really excited about that. In fact, in the studies in Italy, they showed on average, and they're using actually direct from blood culture tests there in these studies, they were still getting a 25-hour faster time to result for the T2 result. And in fact, in the longest case, they're getting 95 hours time savings. A lot of enthusiasm from the 2 sites that did this in order to identify for them, it was the KPC patients that they cared about in their institutions. Knowing that they have a patient that has a high likelihood of being infected with a KPC-resistant pathogen can cascade into a lot of things, like quarantine of the patient, different therapy, and so forth. Shift gears to Candida auris. We were approached a few years ago to develop the Candida auris panel by a site in Europe and also by the CDC. So we put this panel together. A nice publication came out of Mycoses, where CDC validated it for use for patient skin swabs, which are in use today to evaluate patients for colonization. You've probably seen the press about Candida auris. It's a significant problem outside the U.S., and it's a growing problem inside the U.S. It's shut down an ICU for several weeks in England. There's a concern about monitoring these patients. The challenge in monitoring today is Candida auris is a slow grower, so when you take that swab eluate, it's a 14-day process to get a positive. And so the T2 Candida auris panel can provide that result in that 3.5-hour turnaround time. And the last panel I'll just touch on is T2 Lyme. This is not related, obviously, to sepsis, but it's an area that we've had some interest in, and we have currently an ongoing multi-year clinical study for T2 Lyme. And our initial study was designed just to look at patients with a rash, and those are patients that have evidence of Lyme disease. We've actually added a second arm based on our conversations with the agency and with the market. It includes patients that don't have a rash but have signs and symptoms of Lyme disease. And so in the first arm of the study, as patients with the RAS. We've released some of the data. And in that case, we're comparing the gold standard here is, and the big question in Lyme disease, how do you know when the patient's infected? In this study, we're using a tissue biopsy from the rash to define that your patient truly has Borrelia infection. And when comparing the different diagnostic tests to that, the blood test that's T2 Lyme based had the highest overall percent agreement, and since PPA and MPA, PCR from the tissue sample was just behind that. And you have your seroconversion and your two-tier test, and then blood-based PCR had a 0% positivity rate. So really the interesting combination here could be a T2 Lyme plus your two-tier test. But we have more work to do. We still have this study ongoing. We're pleased with the number of samples we've gotten, the patients we've gotten, and we're doing our analyses. One of the exciting things for me as a test developer with this test is Borrelia does not live on surfaces. And so we don't have a challenge with ensuring that there's no contamination when you run a sample. And so our specificity is literally 100%. We don't see false positives, even though we have a very high sensitivity. We've done some head-to-head comparisons to direct from blood PCR tests for Lyme disease. In patients suspected of Lyme, we're seeing a much higher positivity rate. And I think that's just indicative to the different limits of detection, as we've already talked about, for T2MR-based testing versus your standard PCR testing. So just to bring things to a conclusion here, so hopefully you're walking out of here with a conclusion, okay, these panels, so T2 bacteria, T2 Candida, both of them are novel and new. Bacteria like Candida opened a new product code with the FDA when we got that clear. These are your first tests available for use clinically in the US. It's direct from blood. We've got the data, we've proven the technology and the ability to develop these tests, and now we can continue to advance and add menu to the T2Dx and then bring a fully comprehensive panel to the market as well. So it's really incumbent on us at T2 and also on the people that we partnership to demonstrate how a direct from blood test should be best used clinically. And we're excited to be seeing such initial exciting data from T2 bacteria that's mirroring the great data that we have from T2 Candida. A couple other takeaways. You have a non-optical detector and a protocol on the instrument that really is robust against interference, robust against the challenges that a direct from blood test has. I didn't have time to go through all the economic data that's out there. There's two really recent studies that came out really demonstrating the economic models for direct from blood tests. And we do that also, we partner with folks to build those models. So thank you for your time and attention. We'll welcome any questions, and please come by and visit the booth.
Great presentation. I have a question about the de-escalation for negative T2 tests. Since T2 bacteria doesn't cover all pathogens, when would it be medically justified to de-escalate based on a negative T2 result?
Yeah, so what we're seeing selective de-escalation for coverage for specific pathogens.
Like if somebody has a catheter in or something and you're looking for staph or...
For staph, so for example, if your epidemiology is such that you're using vancomycin to cover the risk for staph, you come back staph negative, then you're de-escalating on a selective basis. Okay. And then we're seeing that there's particularly the folks on the antibiotic stewardship committees and the IAD pharmacists are the ones that get really excited about that because they're trying to control overuse of precious antimicrobials. Sure. You're not seeing a global de-escalation. And in fact, I think the debate's going to be, even after a panel that detects 99%, whether or not that's the right thing to do, and we're going to have to get clinical studies to show that.
Has anyone run the bacterial detection and resistance detection panels simultaneously? Is that possible? And are the results consistent with each other?
So yes and yes. So the instrument is random access. You can run them simultaneously. In the Italy study, they were using them in conjunction. And what we're hearing from folks, we've got a lot more market research in Europe because of the upcoming launch. About half the people are saying they're going to run them together or run resistance as a reflex to bacteria, but about half of the folks are saying they're just going to run resistance out of the gate. Great. Everyone's looking at me anxiously. You're free to go. Come on up, and if you have more questions. Thanks for your time and attention.